This is module 7.E. It's day 7, not day 6. Don't panic. We have some time here for lightning talks that people have signed up for. But before we get started, just as a reminder, this session, like all other sessions in the workshop, is being recorded and live streamed and will be available on YouTube for further viewing, just so everybody is aware that the session is being recorded. So I've already gone through our list of hackathon details and links. Again, this has been sent out by email and will be available in these slides, and we'll be sending out a more detailed email later tonight about what the schedule is going to look like for this coming week in terms of meeting opportunities. And then we'll also include all the links from today, as well as links to today's presentations in that mail tonight as well. So again, if you haven't yet, please do fill out the hackathon interest survey. If you have a hackathon project idea that you have not shared yet, please do add it to the hackathon teaming document. And so if you'd like to give a lightning talk, today was the last day during the workshop to do that, but we will have an opportunity during the teaming meeting this week, and then the first hour to hour and a half, depending on how many people want to speak, will be allocated at the beginning of the hackathon on Friday for people to give presentations of anything that might come up during this week. Or if you would like to represent, if you make progress on a project this week and you'd like to present again on Friday, that's perfectly fine as well. So for today's lightning talks, we had 5 people who were signed up. Today, then we will get started with Deepa, and if Andrew joins us in the middle, we can have them go at the end.
Hello everyone, I'm Deepa, and the idea that I have for the hackathon is modeling cell-to-cell communication networks in pancreatic islets. I'm a postdoctoral research associate at the Indian Institute of Science. Okay, so I'm interested in modeling islets in the pancreas because it has different, the cells in the islets, there are different types, and the architecture of the cells is also different. And for instance, in rats, we see this kind of an arrangement, and in human, it's heterogeneous arrangement. And another interesting thing over here is different flow patterns that are observed in the islets. The blood vessel could flow, the blood flow could be from the periphery to the center or center to the periphery, and depending on the order of blood flow, whether it's from center to periphery or periphery to center, the communication between the cells also differ, and this is observed experimentally. And I'm interested in setting up a system to model different arrangement of the cell type and different flow patterns. This more or less the long-term goal of the project, and the outcome that I would like to see after setting up the modeling experiment is insulin secretion, and how different structural arrangement of the cell types or the blood flow pattern has an influence on the insulin secretion. And why am I interested in islets? Because this is important in the context of disease states such as diabetes, where experimentally it's known the density of the vessels or the cell mass decreases, and it also has a major play in islet transplantation in diabetic subjects. Okay, so why do I need quantitative modeling? I would like to set up different architectural arrangement of the cell types in islets using CC3D, and also vary proportion of alpha, beta, and delta cells. And this is evident in case of disease in healthy subjects where the proportion of the cell types differ, and also simulate different blood flow patterns. So by doing all this, I would like to see the effect of structural changes on the functional response, which can be quantified in terms of the insulin level, which is secreted by the islets. And So what am I trying to predict in the end? Again, in the case of diabetic and healthy conditions, the ion channels in the cells could be tuned accordingly. For instance, the transfer between the cells can be varied in the context of a healthy subject and a diabetic patient who would be taking some medication. So the ion channels respond accordingly in different contexts. As a result, the insulin response also differs. So these are the things which I would like to predict. And my skills are the following. My PhD was in computational biology. I have some expertise in kinetic modeling, Python, MATLAB, and I've also worked a lot on single cell modeling, SPML-based modeling. And from my teammates, I would like if someone is comfortable with Python. I'm open to learn along with others in the team. So if someone is interested in the project, please contact me on this email address. And these are some of the references, some of the existing modeling studies which are there in the literature. And the last one over here is the title of my work on single cell beta cell model. Thank you.
All right, thank you, Deepa. Any feedback from our instructional team on that so far?
That sounds like a very promising project. Definitely, since you have existing subcellular models, that makes it particularly attractive to do that. And it would be interesting to do a little bit of research on what multicellular modeling of islet cells has been done, just to see what's out there, which could be used perhaps as a template or to inspire some of this. But I think it's really a clear project, and you have a lot of interesting background and prior work.
Thank you. Yeah, I'll definitely look into the existing models in the literature. So I've seen some of it which predict calcium response, the synchronous and asynchronous release. Yeah, I'll definitely look into specific models and maybe discuss further next week. Thanks for the feedback.
All right. Any other comments or remarks? Anybody else in the room have comments or remarks for Deepa?
How do you anticipate the changing the architecture of the different islet cells will impact this? What makes you want to look at different cell densities in terms of their known behavior?
Okay, so again, I know from the experiments, for instance, in diabetes, there is loss of cell mass because of which there is drop in beta cell density. And this could have an effect on the amount of insulin that's released by the beta cells. And also, there are also experimental evidences which show the vascular density drops drastically again in diabetes. As a result, the communication is affected between the blood vessel and the cell. The exchange is affected. So that's why I'm interested in these contexts. I hope I answered your question.
Yeah, you did. I also dropped into the chat. If it's helpful, you might want to take a look at it. Cosmic single cell spatial map of pancreas, of human pancreas, using FFPE tissue slices, for what it's worth. I don't know if that would be helpful in modeling at all. It's long gene expression.
Sure. Yeah, thank you so much. I'll look into it. Thanks a lot.
Great. Thanks, Kimberly. So that is time for this lightning talk. Thank you again, Deepa. Next, we'll have Avishek go. So Avishek, you can go ahead and take it from here.
Hello everyone, my name is Abhishek. I'm a postdoctoral associate at the Department of Biological Sciences at Virginia Tech. I'm working with Dr. Jing Chen on mathematical modeling and growth-induced sliding motility. So these videos that you are seeing over here are very important. So over here, you can see a bacterial chain starting, and the cells are gradually dividing, pushing the other cells in space. And as the cells are being pushed, remember that they are in a medium. So when the cells are being pushed, they're experiencing some kind of drag forces. And due to these drag forces induced and the friction encountered on the cell due to the cell-medium interaction, there is stress developing inside of the cell. And if you notice at this point, when the stress develops significantly, there is a critical stress level after which the bacterial cell breaks into two. So, and what happens after breaking? It is rather interesting. So after breaking, we can see in the second video that two chains are sliding against one another, and they're keeping in touch. On the third video, we can see a group of a colony of bacterial chains moving, and the interesting and the dynamics of the individual chains when they are in a colony is a different story. And sometimes they stop, sometimes they start growing. And one of the important features that I would like to point out from this group video of this bacteria is that individual cells are not rigid. For example, this cell has a kind of like buckling feature. So, yeah. So what are the main, what are the biological background? So we are modeling rod-shaped cells which maintain end-to-end linkages and undergo cell division and growth to propagate the chain, and chains are pushed outward, and that as they kind of like grow, the tips of the chain accelerate, stresses build up of the chain, and there are two existing models. One is discrete rod, a rigid rod model, where we consider the cells to be rigid, connected with nodes, and we have this different kind of buckling patterns from that discrete model. It is a 1D cell model, and I have developed a 2D hyperelastic model where you have a continuous chain and it can buckle. And I currently am working on developing a full 3D model, but it is very challenging. So motivation and why do we care? So we are specifically focusing on a gram-positive bacteria species known as Clostridium perfringens, and it has a high, like it can cause gas gangrene, and those are severe, can cause 100% fatality. So what is the long-term overarching goal? Is developing a mathematical model to understand growth motility mechanisms, which will help us with disease control and utilization of bacteria. For example, we can use the bacteria for drug delivery. So what are the modeling goals? And why do I need quantitative modeling? Because there are gaps in the experimental data. We cannot measure forces or stresses. And as a result, we do not have a clear understanding, and modeling will help us fill this gap, predicting forces based on fundamental physical laws. What am I trying to predict? The overall dynamics of a growing cell chain resulting in buckling modes and the collective behavior when they're in a cluster and in a colony. So here are some of my long-term goals. You start with a straight chain. So when a straight chain grows, it stays straight for a while and then buckles. And when they are in a straight chain mode, the stress profile is parabolic and vanishes at the tips. The velocity profile is linear. Chain buckles to minimize energy. And change in interaction after a single chain breaks into two and slide against each other. And there is a collective motility behavior of colonies of bacterial chains sliding against each other. What can we do with it?
Abhishek, 1 minute.
Okay. Determine critical stress, collective behavior, simulation results to gain qualitative and quantitative understanding. So why CC3D? Because there are limitations in the discrete and the hyperelastic model. And there are several advantages for the CC3D model. And there are some inputs and outputs that I will not go into. So, and that was a long-term plan. We have a short-term plan just for the hackathon. We will start with a one rectangular cell with a fixed height, create a dictionary. And there is one main challenge: make the chain grow exponentially in one direction and keep the height of the cells unchanged, and check if the length is following these rules, and the velocity profile is and the potentiality impedance. My background is on applied mathematics. And modeling skills include some fluid structure interaction, and there are my program skills. What skills do I need from others? A little familiarity with Python coding, Any previous experience with CC3D would be great, but if you don't have it, it's good. We'll learn it together. If you're interested in a short adventure into world of bacteria, you're most welcome. And if you are willing to continue working after the hackathon, please let me know at your earliest convenience. I'm aiming to work on CC3D model and publish it by the end of December this year. These are some resources. For instance, thank you. I know it was a little fast, sorry.
That's okay. Thank you, Abhishek. Any quick feedback from instructors?
I put a couple of comments in the chat. Peter has one. I would definitely start by looking at that model of bacteria that Andreas Deutsch did many years ago. And I'd make your bacteria into compartmental cells where you use links to define the bending modulus of the bacterium. And it's pretty easy to have growth where when the cells grow to a certain size, you split them and add links to keep the mechanical properties of the bacterium the same.
Yes.
And there was a student a few years ago who did a similar model without, and cell division you'd have to do by hand, but it's not hard. You simply redefine the identity of a couple of the sub elements. There was a student a few years ago who did do rod-shaped bacteria with forces and showed how they could co-align when they swam. And so those are all, I think, possible to do in a couple of days and would be nice examples of what's possible with CompuCell, and also might be a good framework that you could then extend to do the things that you've talked about. So I think it's pretty realistic. If you really want very detailed fluid dynamics interactions, and maybe doing it in something like Tissue Forge where you can do the explicit fluids a little more would be interesting. But I think for the hackathon and for the next few weeks, you could definitely do quite a bit in the CompuCell environment. The one question I had was for the particular bacterial species you're working with, when you showed the pictures, it looks like they're confined to a very thin layer of fluid on a 2D surface, or do they have the ability to crawl on top of each other?
They actually do not crawl. So so that is a very interesting question. So crawling mechanism does not exist. What they can do is collide with each other, and they can, like, if two chains collide, then this chain can go on top of one another, and this can be like normal to the plane of the medium. That can happen.
So when I said crawl, I didn't mean literally that they were crawling. They may use type 4 pili or some other mechanism to move, but some bacteria really stay attached to the substrate and don't glide on top of each other. Others can move on top of each other. And so you have to think about whether you need to do a 2D, a 2.5, or a 3D simulation. To start with, in 2D, but it wouldn't be hard to extend to 3D.
Yeah. So that is a very good question, and there are experimental results suggesting that the bacteria, even it forms a kind of like biofilm or something like that, it is very hard for the bacteria to go out of the plane of the substrate because there is a slime covering on the top of the bacteria. So the addition to the substrate and also the slime layer provides resistance to movement normal to the plane of the medium.
So you
and
could,
that is a much greater force than moving in the medium. So they specifically buckle in the field of the medium, but not normal to the field of the medium. Yeah.
So hello everyone, my name is Yasmina Zenadine. I'm a PhD student at the University of Virginia, and for the hackathon, I would really like to model skeletal muscle disuse atrophy in microgravity. So a little bit of background on skeletal muscle. It's a highly dynamic and sensitive tissue, especially to mechanical load, and it can change pretty rapidly within a couple of days to a couple of weeks. So in the presence of mechanical load, you get muscle hypertrophy, like when you go to the gym. In the absence of mechanical load, you get muscle atrophy, so you lose muscle mass and muscle strength. And the response within muscle is fiber type and muscle type dependent. So you have three main fiber types in muscle. You have type 1, which is slow twitch, which provides you with a lot of endurance. You have type 2A, which is a hybrid fiber, and it gives you both power and some endurance. You have type 2B, which is fast twitch, and it gives you explosive power, but very little endurance. And as an example, the soleus is the most responsive muscle to microgravity, and it exhibits the most amount of atrophy. So if you can see on this figure, in this figure on the right, the left panels show the extensor digitorum longus muscle, which is very fast twitch. And you can see that after a few weeks in microgravity, there's not really a lot of atrophy happening in the muscle fibers, whereas in the soleus, in the three panels on the right, you can see there's a big difference between the ground control and the flight muscle fibers. One, you see smaller muscle fibers in general. You can also see this slight transition from type 1 fibers to more fast twitch fiber phenotypes, such as 2X and 2B. So two very well-known regulators of muscle mass are IGF-1, which is a growth factor, and TNF-alpha, which is a cytokine. And IGF-1 is secreted in response to mechanical load, and it's anabolic. And TNF-alpha is released more during disuse, which is catabolic, and it's more of like an inflammatory modulator. And these both trigger the mTOR pathway, which is very complicated, but I won't go into it. But what you need to know is that IGF triggers protein synthesis to increase muscle mass, and TNF-alpha triggers protein degradation, which decreases muscle mass. And this balance between synthesis and degradation is what controls your muscle mass overall. So the motivation for this problem is that astronauts on long-term space missions face significant muscle atrophy, losing up to 30% of their muscle mass, becoming weak, imbalanced, and physically impaired, which can really affect their ability to complete their missions. And if we have a better understanding of skeletal muscle response to microgravity at the cellular and molecular level, this can really assist us in countermeasure development and eventually help not only astronauts, but people who are affected on Earth by disuse atrophy, such as those on bed rest, recovering from volumetric muscle loss, neuromuscular disease, and the elderly, who are a pretty sedentary population. So the goals of the model and why CC3D would be helpful. It would provide more spatial and temporal dynamics information for the muscle cells and cytokines, as well as other native cells to the muscle. That would be difficult to measure in vivo in microgravity because it's hard and expensive to get samples into space. We're going to try to predict the degree of atrophy and how cell behavior is impaired in microgravity. And this is leading up to a bigger project where I want to predict the effects of chronic disuse in muscle on regenerative capacity of muscle. And my hypothesis is that microgravity induces muscle fiber type dependent atrophy that is partially modulated by the immune system and mTOR pathway impairment. And again, the simulation results will provide insight into how the regenerative capacity of muscle can change in atrophied muscle. So as for the project plan, this is an outline of what I intend to do. And if you have any feedback, please let me know. I would like to import a PIF of a skeletal muscle cross-section from a murine histological image. I'd like to initialize cell types like macrophages, satellite stem cells, and fibroblasts, create fields for IGF-1 and TNF-alpha. And then I want to have an external potential force of gravity that can be changed over the course of the simulation. And I was thinking that this would act in the Z direction, so the cells wouldn't actually migrate in response to gravity, but the load that they feel could still affect the cell behaviors. We'd create parameters for volume, secretion, absorption, proliferation, and apoptosis, and then run the model, and we can use maybe a slider in the player to change gravity values and watch how the cross-section changes over time. Ideally, this would be a time course of like 2 to 4 weeks, maybe more, maybe less, depending on how it goes. Here are the resources for this project. So my lab previously made a disuse atrophy model in NetLogo. And if anyone attended Megan's talk earlier this week, she made a muscle regeneration model in CC3D, which is really great. And some background on skeletal muscle response to microgravity and just a NASA report on muscle function microgravity can be seen in those links. As for potential teammates, I have a really strong biological background and space health background. I know some MATLAB for data analysis and basic Python. So I'm looking for people with strong Python coding skills, basic biology background, and any interest in muscle physiology or space health. Thank you.
Great, that's exciting. How much of Megan's material would carry over into yours? In other words, could you use her model as a template to build off of, or are the pathways so different that they don't help each other very much?
Well, the thing is, for the regenerative part, I could definitely use her model as a template but for the mechanical load response, she doesn't really have anything that connects to that. And she doesn't have IGF-1 in her model at all. So that would be building from scratch. But stuff like importing the PIF, I could definitely use her model as like a building block for that.
So I wonder, to begin with, whether you really, maybe you do, but do you really need to model the mechanics of loading? You mentioned that the main effect of loading and unloading is to change the mTOR and ultimately mTOR, but through IGF-I and TNF alpha expression. And so could you start out by having a scalar variable, which is the amount of loading or the frequency of loading, and have that as an input to your IGF-I and TNF alpha pathways?
Yeah, that would definitely work, but I was looking to make like a field because ultimately I want to connect this model to a finite element analysis model and import the loads from there. So if there's like a template for that already, that would be more helpful in the future.
Right. Well, I think so. You could have the load be represented as a scalar field if you wanted, and then the individual cells could read that load, and you have that as an input then to their IGF-I and TNF alpha.
Mm-hmm.
You know, I guess I mean I understand ultimately where you want to go with this. I'm just trying to think of what the sort of the most achievable first steps might be, so it isn't too hard from the beginning.
Right, right.
And so I think that the ultimate vision is very compelling. I'm just trying to analyze it into smaller steps that might make it more practical to get started on it.
Yeah.
Okay. Other comments, questions? Another exciting talk. Pedro is interested, so.
Yeah, can you go back to the pathway?
Oh, yeah, sure. One sec.
Yeah, that's interesting to me for personal reasons, I would say.
Yeah, Pedro's joining us from the International Space Station, so
Yeah.
Joke,
No,
joke.
it's I was trying to link this to personal experience, because sometimes I am an active person, and then sometimes I have to spend like 1 week doing nothing, like this week. I mean, doing the workshop, but not like exercising as I should.
Mm-hmm.
And then I start feeling muscle pain and joint pain. And
Mm-hmm.
I was wondering, for example, is TNF alpha was the main ingredient to that sudden change in health, in life quality. And then it's as soon as I start exercising back, like it takes hours,
Mm-hmm.
the pains disappear completely, and it's as if nothing happened.
Yeah,
It's
there's a pretty big
amazing.
link between the immune system and mechanical load. So it's been shown that exercise can reduce inflammation, but as we see, like in these types of studies, disuse can increase inflammation. So people who are more prone to like inflammatory diseases could have like increased episodes if they're not active.
One thing that my doctor told me once is that when you exercise, you increase a lot blood flow, and that would be one of the main drivers of reduced inflammation.
Mm-hmm.
Do you intend to model blood flow or something?
So I was planning to include capillaries, but not necessarily blood flow. And the capillaries would just be like sources and sinks for cells and secreted factors. But I'm not sure if I'm intending to do like specifically like blood flow in the future model.
No, I don't mean modeling the blood like the fluid, but the effect that that recruiting fibers has on increased flow as a means to reducing certain to like washing the tissue
Oh,
specifically.
I haven't thought about it, but that sounds pretty interesting.
Yeah, because in my intuition, I think that blood flow would be one of the main factors.
Mm-hmm.
Um, but yeah, I mean, I should. Can you send me this network?
Yeah, I'll send you the paper that got it.
Yeah, I would love to stare at it for like 15 minutes, and I better not use this your talk time.
Yeah. So while Pedro stares at the network for a
Pedro,
while.
maybe you could set up a one-on-one meeting next week to go over these and start roughing out
Sure.
some of the ideas.
Yeah, that'd be great. I'd love to join in on this too. This project's pretty interesting to me as well.
Yeah, that's interesting. Yeah.
I would love
Um,
that. Thanks, guys.
Yasmin, are you familiar with Dr. Millie Hughes Fulford's work at UCSF? She's passed away. She passed away in 2021, but she was the first female. I put it in the chat. She's the first female scientist to do experiments in space. So
Mm-hmm.
she has experimental data, inflammation related data, mechanical unloading with zero gravity. So I listed her publications from ResearchGate, and as an example, like one with inflammation, cell adhesion molecule, gene expression. So I want to take a look.
Thank you so much.
Yeah, I happened to be an intern with KQED Quest, and we got to interview her. So that
Oh.
that video is actually I was there. It's kind of cool.
Sweet.
I'm also interested in microgravity and its effects on cells. I wrote a paper in 2006 about it. So I'm looking at the contraction of cells, and it's the cell cytoskeleton that contracts. And I'm looking at epithelial tissue. My name's Susan Crawford Young, and I'm in biomedical engineering at the University of Manitoba, and I'm a grad student there. My background is a contraction of one cell leads to contraction of other cells. And if these contractions continue long enough, this leads to forces on the nucleus, which can change gene expression. And this is a look at epithelial cells and how they're tied together. And it's the apex of the cell, and they have an actomyosin ring around them. So this is a side view of it. And here's the actomyosin join between each of the cells, and there's actomyosin ring around the cells. And of course, it's attached at the bottom to the extracellular matrix. And what I'd like to change is that I'd like to make the upper part of these cells contract so that this area becomes smaller and they become a tall and columnar cell. I just saw someone has already done that type of simulation, but I would like to concentrate on the cell cytoskeleton. This is an example of a contraction event where the cells are contracting and the contraction moves across the surface. And you can see it starts with one cell and then moves from there. So the hypothesis is the cell contractions that last long enough for a force to be sustained in the nucleus will change gene expression. And this is thought to be a link between the mechanical force and genetic expression. So I would like to model this. I'd like to model how to get a force into the cell, how long that force lasts, and how it affects its neighbor cell. This is mostly how a force on one cell moves to the next cell. And there's basically a phase transition when you go from a flat cell to a more columnar cell because there's a change in cytoskeleton configuration. And the cells have a refractory period. In other words, after undergoing a contraction, they can't sustain a contraction after that for a period of time. So the contractions will move in a certain direction and won't move backwards. And I saw this and I went, yeah, that's basically it. You've got cells and you perturb one cell and stretch it out and see how that affects the neighbor cells along the line. So basically I want epithelial cells on a substrate, and the inputs are a buildup of calcium and an actomyosin buildup, maybe in the cap of the cell. And then you have a cell cramp on one cell that makes the top area smaller and it becomes columnar. And then how does that affect the next cell? So the model would be an epithelial layer, one cell deep, and place the attachment forces at the apex of the cells, where there is actually a trijunction usually in the flat 2D model, and then model the cell cramp. And these are some resources. I was with the group that's been looking at this for a very long time, since the 1990s. And there's the most recent paper here in 2024, and I've included it because it has some explanations of these cell cramps, and they call them differentiation waves. And potential teammates, my background is electrical engineering and education, actually, and I have some biology. It's mostly embryology, and I've been studying mechanics as a grad student. So for my teammates, I could use someone knowledgeable in Python and in cell biology. So thank you.
Great. Okay. So feedback from instructors. By the way, Susan, my background's mechanical and education, so you're in good company there.
I need more mechanical. Anyway, yeah.
So
Good combo.
I put a little feedback in the chat. There people have used CompuCell to do models where you have stress-induced contraction, usually in 2D rather than 3D. In other words, you assume that with the cell contracts in the transverse direction, it elongates, but to begin with, people tend not to model that because it's more expensive computationally. There was a group a few years ago that did a relatively detailed model of stress fiber generation, where they took the cell, divided into lots and lots of compartments, and put fibers between the compartments, and the lifetime of the fibers depended on the stress on the cell. So that kind of thing is possible. It's a pretty expensive computationally, but it's definitely something you could do. So I think both of and we didn't cover it this year, but I put a link in the chat to a simple calcium wave model that we've built that you could look at. And so I think all of the components are possible. You might find again that TJ's tissue forge would allow you to do, as a framework, might allow you to do a little bit more detail than CompuCell, but I think CompuCell is easier to work with, and it would be a good way to work out some of these ideas. So I don't know whether you can build all of that in 3 days, but I think you could make good progress on the beginnings of it.
Okay. Thank you.
Sounds like Katrina has a question.
Yes, indeed. Out of curiosity, Susan, I'm interested in proliferation, which is induced by stretch in endothelial cells. And you said that the nucleus may sense the stretching or the contraction. Do you have any idea of what receptor this should be, or what would be the signal transduction?
There are some stretch pores in the nucleus. I don't have the reference just handy. But the pores widen during stretch, or some of them do, and so more external cytosol is allowed into the nucleus. I think simply compressing the nucleus does rearrange the molecules in the nucleus, and therefore it does affect gene expression.
Okay.
It's
So-
just as simple as that. It's a simple idea. I don't know the details. You'll have to ask Natalie Gordon. That one.
Okay, Natalie Gordon. Okay, thank you.
Yes. She's in my references there. Yeah, she's written extensively about the genetics of this.
Thank you, Susan.
Okay.
All right, any other comments or questions for Susan?
I was just very basic question. I didn't realize that just epithelial cells, skeletal muscle kind of cell, but just regular cells could contract. When you kept saying contraction, I kept thinking muscle cell. This is not muscle. This
It's
is
a,
non-muscle actin myosin.
yes, it's non-muscle, actin and myosin. And it does contract, and it causes the cells to be in tension. And so mechanically, they're a tensional element. And when they're a tensional element, you can call them a tensegrity, and they behave differently from a normal structure because you push one part of a tensional element and the whole structure changes shape. So it's
Gosh.
interesting.
I always just thought that those kinds of cells, that structure was just by kind of microtubule structural elements, not by. Maybe I'm
Oh,
forgetting my physiology from years ago, but
yeah, well, they have actin and then actomyosin, and the actin and the microtubules are attached to each other, and that creates a tensegrity object. If you look that up, because you've got basically rods and strings attached to each other in a structure. And then if you add myosin to it and some calcium and some ATP, well, it moves.
sounds like a great problem for model.
So we'll have one last question from Eurene, and then we'll go to lunch. Eurene, it looks like you did what I did a second ago and clapped while raising your hand. The emojis are too similar.
Yeah. I don't know if I missed something at the beginning, but you said the epithelium, it contracts. So like what happens? It contracts and then it expands, and then I don't understand what happens.
Well, it contracts, and then I guess if it runs out of calcium, it relaxes like any other, like a muscle would. It's a similar mechanism to what happens in a muscle, but it's non-muscle myosin, but it still acts in a similar manner. And then
So sorry.
it rearranges the cell cytoskeleton, so it tends to be more of a columnar type cytoskeleton. Anyway,
then it goes back?
theory. It'll flatten back out, yeah.
So this happens in all epithelia, that it has this periodic contraction and then expansion. I don't know if I missed something.
Yes, I would say in all epithelia and all living things.
Does this serve some kind of purpose?
It is cell differentiation mechanically. Mechanical cell differentiation. One way it'll.
But then you mean the epithelium gradually transforms to different types of cells?
Yes, you can get a nice flat epithelium, say on the surface of a salamander egg, because I know about this. And there's a contraction wave that goes across the surface of them. And the theory is anything that's been touched by that contraction wave turns into a neuroplate, which is a more columnar type cell. And then it rolls up and forms the brain in the spinal column.
Okay, yeah, that was one question. I actually had a second question, if that's okay. You said the contraction of cells leads to contraction of other cells?
Yes, it
Like
tends,
how?
oh, well, that's why I wanted to model it. You contract one cell and it's going to pull on the one next to it. So that's pulling on it. So it's stretching it, so it reacts and it contracts.
Is it active or?
Oh, they're active, yes. Like I
So it's
said,
an active response.
yes, yes.
Yeah, I'm also interested in modeling epithelia, but maybe not the same kind of effects. I'm actually kind of interested in your project. I'm more of a theoretical physics though, so maybe someone else with a
Oh, no,
more biology
theoretical
background could join.
Sorry, theoretical physics is great. Helps solve problems.